Thank you very much, Dr. Nestor, for that very kind introduction, and thank you, Dr. Baird, for the invitation. It's really a pleasure and a privilege to get to talk to you today, and thank you for coming despite the snow. And what I'd like to do over the course of the next 45 minutes or so is not only tell you a little bit more about what I do, which is trying to understand new pathways of inflammation, but I'm going to craft something a little different from what I normally do, which is to try and emphasize how I think it's important to lab medicine in the future, how not only the lessons that we've learned, but also the approaches that we're taking are, I think, quite relevant. And I'm going to start by framing the problem, which is an issue of immune homeostasis, and try and tell you why I think it's important to study it, tell you a little bit about how we study it, using small molecules as tool compounds to perturb the system. And this is going to lead to two vignettes and two little stories. I'm going to tell you about two new regulators that we found. They're both kinases, two kinases called DYRK1A and CDK8. And wrap up, like I said, about telling you about why I think this is relevant to lab medicine. So the immune system faces a fundamental challenge. It's got to respond very aggressively against pathogens, and at the same time, it needs to maintain tolerance to self, right? And we understand that it does this by executing a careful balance of pro and anti-inflammatory forces. Excessive perturbation of this balance in either direction results in disease. So too much inflammation leads to autoimmunity, too little inflammation leads to infection and predisposes to cancer. And these are obviously big ticket items. They're really important diseases. I've highlighted the size of this problem in the United States with these numbers, and I've cribbed from the CIA World Fact Book to try and give you the point that these are country-sized problems. These are big problems. So if you look at autoimmunity, it affects over 50 million Americans, and direct medical costs alone exceed $100 billion a year. And this does not include the indirect costs, which are thought to be much higher, because these are chronic diseases, diseases with a lot of disability, lost ability to work, so on and so forth. Cancer needs no introduction, second biggest killer in the country. The point is these are big items, important problems, and immune homeostasis is an important factor at the center of it all. And so to try and understand new factors that regulate immune homeostasis, to get new tools to modulate immune homeostasis is a really important endeavor. Immune homeostasis is maintained by many different cells collaborating together. We've focused on the CD4 positive T cell, which I would argue is a key quarterback of this balance. And the CD4 positive T cell can differentiate into either pro-inflammatory subsets like Th1 and Th17, or anti-inflammatory subsets, largely represented by regulatory T cells or Tregs. And in particular, we've focused on trying to understand more about Treg biology because this is the dominant player on one side of the curve. And so we reasoned that what we learn about Treg biology is generally applicable and relevant to many different diseases. There's lots of data out there to tell us that Tregs are important. I'll just tell you some lessons from genetics. So if you look at Mendelian diseases, patients who lack the key Treg transcription factor FOXP3, they develop IPEX syndrome, lethal autoinflammation by age 5. If you look at complex human diseases, you see a consistent theme, which is an overrepresentation of genes that we know are important for Treg development and function. This is an example list from ulcerative colitis genome-wide association studies, but you see the same lesson whether it's type 1 diabetes, pick your favorite autoimmune disease. Disease genetics, bottom line, are telling us that Tregs are really important at regulating inflammation. So what are the tools that we have to this date? This, I think, represents what I think of as some of the best leading-edge tools that we have to modulate Tregs specifically, which is low-dose IL-2. I remember when this was published, back-to-back papers in the New England Journal. It was really exciting. And while there is reason to think of why low-dose IL-2 is relatively specific for Tregs, you'll recognize that IL-2 is a really pleiotropic cytokine. Hits many different cells, certainly not just Tregs. And I think this highlights a more fundamental issue, which is to this day we still don't have enough tools to specifically diagnose or treat defects in T cell differentiation, whether it's Tregs or pick your favorite subset. And to develop those tools, we're really going to need a better understanding of the underlying biology. So that's the effort that I set upon probably about 10 years ago. So the bottom line is we wanted to identify new pathways that regulate Treg differentiation and how we might be able to perturb them. And the way we went about this was using a small molecule screening pathway. And the idea was that if we could find new molecules that enhance Tregs, we could use these as tools, for example, to try and ask, what do those compounds do? How do they work? What proteins do they bind? Can we learn more about the biology? Can that biology lead us to new clinical and therapeutic hypotheses? And in fact, I'm going to highlight two of the vignettes of two different regulators that we've identified in this way, DYRK1A and CDK8. The second motivation behind this was whether or not we understand how the compound works, we have a compound that has an effect of interest, right? And can that potentially be the start of a drug delivery program? And finally, I'm going to allude to some lessons as we've learned about so many different new molecules, about our ability to think about these in classes of mechanisms, and how that leads us to think about new lessons in drug repurposing, as well as a step towards precision medicine. So this slide really captures the heartbeat of a lot of what we do. So fundamentally what we do is we take naive CD4 positive T cells, we do it both from mice as well as humans. We differentiate them in carefully calibrated conditions. So I've highlighted some of the examples down below. So we basically can differentiate towards all the major T cell lineages. We carefully manage the dose of each individual relevant cytokines. And this allows us to do two things. We can identify suboptimal concentrations, shown here by the blue triangle. It's really good at identifying enhancer activity, so we can quantitate that. And we can identify near optimal conditions, shown by the red triangles. It's very good at quantitating inhibitory activity. So the point is, what we've done is we've turned what is classically a very artisanal, a very variable assay into something that is scalable and semi-quantitative. Okay, so this is representative data from 384 compounds, and we're looking at the ability to enhance Treg differentiation. What I'm trying to show you is in blue, the negative controls are very well separated from the positive controls in red. And this is the key to the whole success, which is most of these compounds you expect to have absolutely no effect, and indeed they cluster very tightly around the negative controls. That allows us to highlight a small, reasonable number of candidate hits above the dotted line. These would be the enhancers that we pursue further. They're highlighted in purple. So I'm going to start by telling you about our pilot study. This was an interrogation of 4,000 small molecules, largely FDA-approved drugs and tool compounds of known interest. From this library, we were able to identify 20 compounds that specifically enhance Tregs. Now, the specific part is important because if you imagine, if you want to treat autoimmunity, you might want to boost the anti-inflammatory Tregs. You don't want to boost the pro-inflammatory cells. So we go back with all these 20 compounds and we specifically interrogate their effect on all of those lineages individually. So what you're looking at here are basically, for a single compound, you're looking at dose titration effects, increasing dose of the compound as we go to the right. We're looking at the ability to boost Tregs in Treg conditions, that's shown in blue. We're looking at the ability to enhance Th1 or Th17, those are the pro-inflammatory subsets, in red or orange in their respective conditions. So what you want to see, short story, is blue high, red, orange low. And that's true for all 20 compounds. These 20 compounds include all 6 known Treg enhancers in the library. That tells us we're doing a good job with sensitivity. It also tells us we're capturing 14 new compounds. So we've more than tripled our chemical understanding of this space in Treg biology, right? So it tells us there's a lot more left to be learned. We haven't exhausted the pot yet. When you start to learn about so many different compounds, you can't help but start to notice some patterns. And the first thing we noticed is many of these compounds are really cytotoxic. So that's represented here in this chart on the y-axis. This is essentially the therapeutic index. So the way I think about it, lower down, more cytotoxic, higher up, less cytotoxic. I'll point out that rapamycin, which is perhaps the most studied of all these pro-Treg compounds, is amongst the most cytotoxic, cytostatic of them all. And so what that tells us, I think, is there's a lot of room to find new compounds that work better. Going to the right, we have increasing maximal Treg effects. So intuitively, you want things in the upper right-hand quadrant. I've highlighted them in blue. These include some known enhancers, including the statins. I'm not going to talk about them today, but they're interesting, simvastatin and lovastatin. And a compound that I am going to talk about today prioritized through computational methods I don't have time to discuss, which is harmine. So drilling right deep into harmine. Okay, this is showing you that harmine is a really good enhancer of Treg differentiation, not only in murine cells, we're going from 40 to 90%, but also in human cells, we're going from 8 to 22%. It's also got a great inhibitory effect on pro-inflammatory Th17 cells. We're going from over 30% to basically undetectable. So it's got great anti-inflammatory effects in vitro. We spend a lot of time proving that these Tregs are functional. So it's not just that it's expressing a single gene. We want to make sure that they work. And so what we're doing here is we are comparing these harmine-enhanced Tregs in blue to the gold standard in the field, which is TGF-high-driven Tregs in red. And if the only thing you need to take away is that blue looks like red. This is a standard in vitro suppression assay, increasing Tregs as we go to the left, giving us increasing suppression of co-cultured responder T cells, and the point is blue tracks red. We have 3 animal models of inflammation, so this is a lot in the field. These are all T cell transfer models in different organs, different specifics of immunology I can discuss. But the short story is without any Tregs, you get a lot of disease in the individual organ. That's shown in the black line. Lots of inflammation in the colon, rapid onset of type 1 diabetes, lots of inflammation in the airway. Transfer of either harmine-enhanced Tregs or TGF-high-driven Tregs gives you significant and comparable attenuation of disease, right? So you have a lot less inflammation in the colon, you have delayed onset of type 1 diabetes, you have a lot less inflammation in the airway. So these Tregs appear to be fully functional. Okay? This leads us to ask, can we use it as a drug? This turns out to be a difficult question because, frankly, harmine is a lousy drug. It's got a serum half-life of less than 5 minutes. It's a difficult thing to work with. But what we were able to do was play with anatomy here. So what we reasoned was if we could have a prolonged local exposure to harmine, maybe we could have some local effects before it's absorbed into the bloodstream. And so what you can see is if we give harmine by enema, we can see increased local colonic Tregs. If we give it intranasally, we can see increased Tregs in the lung. This led us to try and go one step further. Can we go back to our airway model of inflammation, basically a model of asthma, right? And so what this is, let me just take a second to tell you this is an immunization recall challenge model of asthma. So we immunize mice at day 0 with ovalbumin, and then we wait 3 weeks, we do nothing, and then we come back 3 weeks later and we give them a challenge of nebulized ovalbumin, and without any therapy, you get lots of inflammation, lots of eosinophils in the bronchoalveolar lavage fluid, lots of inflammatory cells around the large vessels of the lung. Now, if you simply give them 4 days of harmine, so 4 doses of harmine at the time of immunization, daily from 2 days before to 2 days after, and then you do nothing, no more harmine, no nothing, you wait 3 weeks and you come back and you challenge them, you basically see undetectable inflammation by both histology or bowel eosinophils. And so this works even better than the gold standard, which is tolerization of ovalbumin, suggesting to us that this is a candidate therapeutic. So that's interesting. We're really particularly interested in how it works. So harmine is known to hit 3 classes of targets: monoamine oxidase, CLK kinases, and a family of kinases known as DYRK kinases. And so we wanted to figure which of those were the most important. So what you're seeing here are dose titration curves, increasing dose as we go to the right, and looking at the ability to enhance Treg differentiation. In all these curves, harmine is in blue. It reliably enhances Tregs really well. It's great to work with. If we use either a compound that only inhibits monoamine oxidase or CLK kinases, you see absolutely no Treg effect. If you use 2 structurally different molecules that both inhibit DYRK kinases, they both enhance Treg differentiation. They both inhibit Th17 differentiation. So this is chemical evidence that DYRKs are important for this phenotype. This is a family of 5 kinases. We go back and knock them down one at a time. We find that the most important is the family member called DYRK1A. Knockdown of DYRK1A, shown in blue, gives you increased Tregs, decreased Th17. So genetic inhibition phenocopies chemical inhibition. It tells us we've identified a new regulator of Treg Th17 differentiation called DYRK1A. So less DYRK1A gives us less Th17s and more Tregs. And then we became really interested in what happens when you get more DYRK1A. Do you see the converse, the opposite phenotype? And why do we care? We care because we think that this is clinically relevant. So it turns out that DYRK1A is on chromosome 21, which is triploid in patients with Down syndrome. DYRK1A is widely expressed in all different cell types. This is data from Joaquin Espinosa's group in Colorado. Each color represents a different cell type, and the point is T cells, which are in purple, express a lot of DYRK1A. And T cells from people with Down syndrome express 50% more DYRK1A than that. Okay, so they express the gene, and it turns out, and I did not clue into this from medical school, but it turns out that patients with Down syndrome have a lot more autoimmunity, up to 100-fold more autoimmunity depending on the study you read. This includes common autoimmune diseases like thyroid disease, type 1 diabetes, and celiac disease. So I'm giving you some example data from the Box study, which looks at type 1 diabetes in the UK, which I think is perhaps the best studied of all these phenotypes. So this is a study of over 2,000 people who developed type 1 diabetes and looking at age of onset. This is typical controls on the right here, and you basically see a roughly even age of onset after age 1. Now, if you look at patients with Down syndrome, not only are these bars higher, they basically calculate a 4.5-fold increased incidence of type 1 diabetes. What you see is a bimodal distribution. So instead of this even distribution, you see a left shift of part of this curve. You see a lot of kids developing type 1 diabetes at a very young age, even within the first year of age, which you basically don't see in a typical cohort. So they get more autoimmunity, they get more aggressive autoimmunity, and we're really interested in trying to understand, is this driven by perturbation in the Treg Th17 axis? So we started looking in some murine models. We've looked at 2 mouse models that overexpress DYRK1A. This is work done by a great technician in my lab, Keegan Moo, who's looking at graduate school this year. And we started by asking, do we see increased Th17 and decreased Treg differentiation? A very simple question. And so the short answer is we do see increased Th17s, like you might expect. This is a complicated graph, but I'll go through it. The bottom line is what we've done here is we've set up some very sensitive co-culture experiments to ask if there are differences and convince ourselves that there are differences in Th17 differentiation. What we're doing is co-culturing either DYRK1A overexpressing cells. Here we have the BAC model on the left and the TS65DN model on the right. We're co-expressing either DYRK1A overexpressing cells or wild-type controls together with a congenic control. So the first thing I'll point out is the congenic control is in green. It's basically just a reporter. We're looking at increasingly pro-Th17 conditions as we go to the right, and the green dots go up as we go to the right. And so what that tells us is there's no cell extrinsic effects. We're looking at co-culturing of the wild-type cells. That's in blue. Blue looks like green. Again, that looks like wild-type. This tells us there's no weird genetic thing going on from the background. Now, if we look at the DYRK1A overexpressing cells, that's in red. The point is red looks like blue. It's more prominent in this part of the curve where you can see basically DYRK1A overexpression drives increased Th17. And we think that this is going to lead to increased inflammation. The same thing in a mouse model of Down syndrome. Okay? Again, here the DYRK1A overexpressing cells, the Down syndrome models, mice, those cells are in red compared to the littermate control in blue. Again, red is higher than blue. It tells us that DYRK1A seems to be overexpression of DYRK1A in this model also seems to be driving increased Th17. What about Tregs? We've done the same kinds of assays. The short story is you'll see all these lines are completely overlapping. If there was a defect in Treg differentiation, we would expect a solid red line to pop down below everything else, and we don't. And so it seems that what we're observing is basically a nonlinear relationship where overexpression of DYRK1A does perturb Th17, gives you increased Th17, but does not affect Treg differentiation. What about Treg function? We're comparing, again, the BAC model on the left, the Down syndrome model on the right. As you can see very clearly, the lines are completely overlapping, telling us there's no defect in Treg function. So we're narrowing down the area that we want to look at. So what about development? in vivo. So we've looked at the mice, and this is going to be a point I'm going to come back to at the end. So we're comparing, again, the back model above, the Down syndrome model below. We're looking at DYRK1A overexpressing mice in red, and the point is blue looks like red throughout much of development. We're looking all the way from the thymus into the periphery. Now, we saw increased Th17 in vitro, but what I'm going to point out here is you don't see increased Th17 in the spleen, but you do see increased Th17 in the intestine. And this is going to highlight a point that the intestine is a naturally rich source of Th17 cells. Th17 is being driven by a stimulus. There's the intestinal microbiota. And so the point is you uncover this drive to make more Th17s in the context of the appropriate stimulus. So the appropriate stimulus is important to uncover subtler phenotypes. What about in humans? So this is really the big thing that drew me to Seattle. At the Benaroya Research Institute, they've had a long and proud history of collecting samples from human subjects, and learning important mechanistic lessons. And so I came here, and they've been really helpful and really supportive in helping me set up a biorepository of people with Down syndrome. We started in April 2018. We've got a great clinical collaborator. She's dressed up as Jessie here. I'm the minion to her left. And she basically has the major Down syndrome, basically the only Down syndrome program in the state. And she sees patients from a 5-state radius. We've been really successful at recruiting. We've got over 100 individuals in the registry. Data I cannot present because the experiment is ongoing today, this whole week actually. And we are doing our first big study of samples from 25 people with Down syndrome compared to controls and people with type 1 diabetes. We're looking at mass cytometry, so we're looking at over 50 parameters simultaneously. We're looking at their responses to different phosphorylation of different STAT proteins in response to cytokines. We're doing RNA-seq, and we're looking at the Tregs. So we're doing a whole lot of work, and I hope to have more to show you the next time I'm here. Our preliminary studies, what we can tell you is that there is increased DYRK1A in the T cells from people with Down syndrome. This just basically validates what we saw before. And when we look at these T cell subsets, we don't see any differences in Tregs or Th17s, although perhaps we're not looking in the right place. Perhaps the right place to be looking is in a gut biopsy, and that's also of interest. We do see increased Th1s. This alludes to the fact that there are a lot of other genes on chromosome 21 as well, including the interferon receptors, and this might be also contributing to the pathology. We're certainly interested in how DYRK1A is giving this Treg Th17 phenotype. If we look at major known targets, one of the important targets is NFAT. So these are Amnis experiments. This is essentially a flow cytometer hooked up to a microscope. Going to the right is increasing nuclear translocation of DYRK1A. So unstimulated T cells shown in black, most of the NFAT is in the cytoplasm. If you stimulate them, it starts to move into the nucleus. That's the red line. If you add harmine, more of it goes into the nucleus. We're interested in, does this help drive the Treg Th17 phenotype? Other candidate pathways, including regulating alternative splicing, we think this is really interesting. There's very little known about alternative splicing in T cell differentiation. In fact, very few of those data sets exist. We managed to cull one of those, and we tried to ask what are candidate genes that are differentially spliced. We came up with one of those, which was Fin kinase, and we asked, does harmine perturb that splicing? And indeed, harmine seems to give you increased predominance of the Fin A isoform as compared to the Fin B isoform. Whether this is, the relevance of all of this remains to be more broadly explored. So to sum up this part, what I've shown you is that stimulation of primary cells reveals important and novel biology. We've been able to use these small molecules as drug-like probes and identify the compound harmine that led us to understand that DYRK1A inhibition enhances Tregs, inhibits Th17 differentiation. DYRK1A overexpression gives you increased Th17 differentiation, and led us to an interest in the immunopathology of Down syndrome. What I'd like to do is to turn for a couple minutes to another vignette, which is a different regulator of Treg differentiation, the cyclin-dependent kinase CDK8. This work done in large part by Aslan Arnett, who's been a superstar technician in my lab. So after we proved that you could actually learn from primary cells at scale, which was initially received with some skepticism, we started to ask, could you look in other immune cells? And my buddy in the lab, Tom Sundberg, was interested in innate immune cells, so he asked about things that increase tolerogenicity in myeloid cells, in dendritic cells. And then he started getting successful, and we started trading compounds by asking, are compounds that are tolerogenic in one cell type tolerogenic in the other cell type? And we thought at first we were really smart in doing this, and we were going to get twice as many papers. And in fact, most of the time what we saw was this particular phenotype, which is things that increase Tregs, for example, harmine here, don't increase IL-10 in myeloid cells. Compounds that increase IL-10 in myeloid cells, this is the pan-salt-induced kinase inhibitor, have no effect on T cells. And then one day, we had a really surprising observation. We'd identified this compound DCA. It's a great enhancer of IL-10 production in dendritic cells. And it turns out to be a good enhancer of Tregs, shown in blue here, as well. And not only that, we've developed a couple of other assays by this point. We're able to assess Th2s. We can show that DCA not only enhances Tregs, it also enhances Th2s in black. And we asked, do all Treg enhancers also enhance Th2? And the short answer is no. In fact, harmine very clearly does not enhance Th2 differentiation. So this is drilling right down to give you high granularity. DCA is developed as an inhibitor of CDK8. It's a great enhancer of Tregs. You're going from about 20 to about 70%, not only in mice, but also in humans. Here you're going from about 15 to 30%. It also enhances Th2, I pointed out, and it also inhibits Th1 and Th17 differentiation, about 50% for each. This was originally designed as a CDK8 inhibitor. CDK8 is the only catalytic component of the CDK8 submodule of the mediator complex. The point is that regulates RNA transcription. It's a big and important regulator of RNA transcription. And so we're trying to ask, is CDK8 the relevant target? So we use, again, chemical inhibition. So DCA is here in blue. So we use a different CDK8 inhibitor that's in green, also enhances Tregs. If we use genetic inhibition, so what we've done now is we've introduced CRISPR-Cas9 deletion in human T cells. So we're trying to get closer to humans. And you see here increased Treg differentiation. So again, genetic inhibition phenocopies chemical inhibition, tells us that CDK8 is an important regulator. We're doing some functional interrogation. Again, here we're comparing DCA-driven Tregs in blue to the gold standard, which is TGF-driven Tregs in red. The point is going to be blue tracks red. Okay. Standard in vitro suppression assay, increasing Tregs as we go to the left, increasing suppression of co-cultured responder T cells, blue tracks red. Two different animal models, I've already shown them before, of type 1 diabetes and colitis. No Tregs, you get rapid onset of type 1 diabetes, lots of inflammation in the colon, transfer of either DCA-driven Tregs or TGF-driven Tregs gives you significant and comparable delay in type 1 diabetes, decreased colonic inflammation. So this is telling us that these CDK8-driven Tregs are fully functional. We're interested in how they work. I'm just going to spend a second on how they don't work for a reason that I'm going to come back to. So CDK8 phosphorylates important transcription factors, including STAT1 and STAT3. So the obvious question was, is this how it's regulating T cell differentiation? What you're seeing here is, and it phosphorylates these proteins not on the canonical tyrosine that immunologists are used to thinking about, but on a serine residue in an activation domain. So we're looking at phosphorylation on this serine residue. You can see with the appropriate cytokine, you get increased phosphorylation. And if you add DCA, you see abrogation of the induced phosphorylation, but not the constitutive phosphorylation. So this is going back down to 1, it's not going back down to 0. So CDK8 does phosphorylate on the serine residue. Is this important for Th1, Th17 differentiation? The short story is, if it were, then what we're looking at here is mice where the serine has been mutated into an alanine. So CDK8 can't regulate phosphorylation on this residue. If this is necessary, what you would expect to see is DCA should no longer inhibit Th17 or Th1 differentiation. It still inhibits Th17. It still inhibits Th1. This is not how it's working. Why am I showing you this? It's for a completely different comparison. What we noticed was something new in the field, which is if you look at these mice where the serine has been modified to an alanine, you see decreased Th17, you see decreased Th1 differentiation, suggesting that this might be an important therapeutic target in the future. I do have some positive data to show you about how we think CDK8 inhibition is working. One of the things that we look at is the kinetics of expression of FOXP3. So if you look at low versus high dose of TGF-beta, that's the cytokine that drives Treg differentiation, that's in black and red. The point is these black and red lines look very, very similar. They're essentially identical for the first 2 days of culture. Then what happens is with low dose of TGF-beta, FOXP3 goes down. With high dose, that goes up. In fact, most of the small molecules look more like the TGF high pattern. Until we stumbled on DCA. DCA gives us a pattern that we've never seen before. What you see is a really big increase in early expression of FOXP3 with a subsequent decline that's very similar to low TGF-beta. What I think this tells us is that there are different pathways that are being engaged. There are early pathways of, we think that there are early pathways of Treg differentiation, late pathways. DCA and CDK8 inhibition is working on the early pathways, but not the late pathways. And we think this has important implications in terms of combination therapy, in terms of who to treat, so on and so forth. We've done some RNA-seq analyses to try and understand what's the important factors being regulated. We look at transcription factors that are concordantly regulated, and we come up with a single hit, which is AP1. This is a family of transcription factors that we'd previously shown through a completely different method, where it was important in dendritic cells and how those are induced to make IL-10. We look back again, and indeed what you see is DCA gives you decreased phosphorylation on the inhibitory site of the AP1 family member CJUN, but has no effect on the activating serine. And so we're really interested in whether this phosphorylation is how it's driving Treg differentiation. So to sum up this part, what I've shown you is that CDK8 has really broad immunoregulatory roles. It affects both innate cells and my adaptive immune cells. It has broad effect on all major T cell lineages. It also tells us about how chemical immunophenotyping may inform us about important lessons. So what I've not told you yet is that CDK8 inhibitors are being designed as chemotherapeutic agents. So for example, for colon cancer. And so I think it's very important to know upfront if you want to treat a patient with cancer that your drug potentially has tolerogenic effects. It may be good, or it may decrease the tumor surveillance. And so, what's going to be the effect on remission? It doesn't mean that that's a bad drug, but you might want to know about it so you can monitor it or use other adjuvant therapeutics. And it also suggests a different role, right? A new role for something that you were going to use as a chemotherapy. Should this be considered as being used as an immunomodulator? We've learned about new biology from DCA showing this regulates early pathways of FOXP3, and what the mechanics of those are of great interest to us. And finally, it tells us about STAT1 and STAT3 serine 727 as a novel therapeutic target. So what I'd like to wrap up this data part is by showing you how we've been able to use screening of small molecules to learn about new biology. I've told you about 2 vignettes about DYRK1A and CDK8 and how that's led us to new clinical and therapeutic hypotheses. DYRK1A led us to be interested in the immunopathology of Down syndrome. CDK8 leads us to not only learning about new biology, but also implications on a potential emerging chemotherapeutic and whether that has drug repurposing options. And I want to wrap up by pointing out some things about why I think this is relevant to lab medicine. Why I would care if I were sitting on the other side of this podium. The first is, I think a really important concept is perturbations uncover important biology. And I can't overstate this enough. So homeostasis is a really powerful force. It tries to bring us all back to a healthy state, regardless of where we want to live. Right? And this can mask important but subtle but important differences. Okay? And so the point is, if we look at steady state, important differences can often be easily missed. And so if I want to go back to remind you about data from the mice that overexpressed DYRK1A. So we had uncovered that there's an increased drive to make Th17 with that genetic perturbation. We don't see that in the spleen, which would correspond to the peripheral blood if we look at patients, right? And part of the reason we don't see that in the spleen, but we do see this defect in the intestine. And the reason I think we see it in the intestine is because of that natural perturbation, the intestinal microbiota driving Th17 differentiation. And so the point is to uncover these important phenotypes, you may need to use the right stimulus. And that may be something we need to be thinking about incorporating. I'm not the first, by far, person to be thinking about this kind of concept. I'm going to highlight a single piece of data from the Human Functional Genome Project. Huge, huge effort here led by Mihai Natea. This is and what they asked was, if we don't just look at how much cytokine lives in the serum, if we ask about production in response to stimulation, for example, with poly-IC or other toll-like receptor ligands, and ask, can we find new relationships? Can we find new genetic elements that regulate this production? And indeed, they were able to find many such relationships only after perturbation. And so here they're finding a new genetic relationship of a new genetic element that regulates IL-6 production in response to poly-IC. There's many other such vignettes in their papers. And so the point is perturbation teaches us new biology, and that's maybe something we need to be thinking about. The second lesson is what I think we've been doing helps us to do something interesting, which is basically map out an immune landscape, right? With all these, if you take away the details of the individual compounds, what we're fundamentally able to do is to ask in a semi-quantitative fashion, does this, whether it's a compound or genetic anomaly, does this have an effect on innate immunity? Does it have an effect on adaptive immunity? What kind of effect is it? Is it making more IL-10? Is it making more pro-inflammatory cytokine? Is it differentiating in different lineages? And you can see we're starting to bin these compounds into different classes, right? So harmine, for example, only affects adaptive immunity. Syk inhibitors only affect innate immunity. DCA affects both. And different patterns of effects on T-cell differentiation into different subsets. And what that's starting to do is to break things down into different complementary groups, right? You start to think about them functionally, right? What kind of a genetic defect does this person have? What kind of immune effect does this compound have? And can we use this to direct the right drug to the right person? So the really simple concept is round peg in a round hole, right? But here we're starting to try and use this to define what is a round hole, right? What's that even mean in immune space? And so we think that this is going to begin to help us define those features. And so the idea is to deliver the right drug to the right patient. The single extension that I will leave you with is, can we think about this in the terms of companion diagnostics, right? So right now, how do we select immunomodulators? I think a lot of that has to do with, is really empirical. Either has to do with, hopefully your physician has good experience, or basically we select them based on what your insurance will pay. But wouldn't it be nice if you select a compound based on its, we now have biologics that have particular precise effects. Wouldn't it be nice if you could test a priori, if you're using a drug, for example, to modulate Treg Th2 differentiation, to test up front that patient actually does respond in the way that you would expect. And so that's the kind of concept of a companion diagnostic that I think is particularly interesting. All this kind of work obviously takes an incredible amount of support. I've acknowledged the two great technicians in my lab, Keegan and Aslan, as we've gone by. Incredible amount of institutional support, not only from Jane Buckner, but also the Translational Medicine group led by Gina and Cassidy. Rebecca, I've mentioned, she's a phenomenal clinical collaborator in all the subjects and their families. My postdoctoral work was done with Ron Xavier. I've mentioned Tom, who did the DCA work in innate immune cells. Lots of support from systems immunology at BRI, and David and Zia and Ye were kind enough to share mice with me. With that, I'd really like to thank you for your attention. I'm happy to take any questions. Yes, sir.
Interesting data. A number of your DYRK1A inhibitors had a kind of a inverted U-shaped curve, a response curve that's curious.
That's a great question. I'll tell you exactly what's going on. Basically, what happens is at really high doses, the compound kills cells. So we've reached the super toxic dose. And so basically, those numbers are meaningless. So what you want to, I know exactly what you're, stop thinking. So basically, there we go. So that's why I think the therapeutic index is really important, because what we find also is it's really kind of pharmacology 101, right? Which is really interesting. So there we go, right? So if we go here, there we go. Oh no, don't think. Basically, what's happening is you get this linear range where you don't have a lot of cytotoxicity, and you're getting increased Tregs. And then once you go past that prime zone, you start killing cells off, and then basically all physics are, these are basically not very informative doses, but we keep them up there for full reference. Sir, yes.
You mentioned the statins being
Yes.
Is there anything you can comment about how this plays out?
Yeah, I think that was the obvious low-hanging fruit that we first looked at. And so if I might be able to put words in your mouth, what you might be asking me is, do statins inhibit inflammation in people? Because obviously we've given a lot of people statins. What's gone on with them? The short answer is we don't really know. So people have looked. I'm not the first person. This was first shown by Ethan Shivak and the Japanese group over a decade ago. And people have looked since then. The data is very controversial. It's very unclear. It's very mixed. But I will leave you with this, which is, how do we know that we're even doing the right study? So we've looked at people taking statins because they take statins for heart disease. This has not been dosed for immunologic effect. So are we looking at the right dose? I have no idea whatsoever, and I don't think any of us do. So we've looked at a dose of convenience because that's what's FDA approved to be given to people. So does this have to do with why we're seeing very mixed effects? So in some cohorts, yes. In some cohorts, no. Certainly, no one would treat lupus with just statins.
Related to that, DRI has a huge type 1 diabetes
Yes.
cohort. Lots of those folks are getting statins. Have you or others at DRI tried to see how that correlates with Treg?
Yeah, so that's a great point. Your prescient point. That's on the docket to do. That's on the docket to do. I think my suspicion is it's going to be hard to tease out, because no one's ever gone back to do the appropriate phase 2 study. Here would be, what dose gives you an immune effect, right? Or are we seeing this pro-Treg effect at a dose that's, are we 10-fold off? Are we 100-fold off? I have no clue. And even more interesting than that, I think, is how are the statins doing that, right? So the idea is that actually it's got to do with a post-translational modification, is the current hypothesis in the field, a post-translational modification called geranylation, and other such words that are really hard for me to pronounce. But the bigger question is, well, what's the protein that's being geranulated, right? I mean, so I think that's the interesting, and that gets back to the kind of glycobiology that, you need a whole different, like you needed to get Vogue again, and you need that expertise to help you try and figure that out. Fascinating questions. Well, thank you very much for your attention.